Hello, my name is Mennwa Abou, and this summer I had the honor of working under Dr. Marie DeMarco in St. Paul's Hospital on a project entitled Using Filter Paper Cards for Quantification of Amyloid Beta Peptides from Cerebral Spinal Fluid. Today I will start with a brief background on my research, introduce my hypothesis, go over my materials, methods, and results, and end with a conclusion. So Alzheimer's disease is a neurodegenerative disorder with no treatment and no cure. Alzheimer's disease is characterized by progressive memory loss and a decline in problem-solving skills, cognition, and judgment. There is no calculated assessment to diagnose AD, so physicians will use a combination of medical history, testing, and the ruling out of causes of other dementia-like symptoms. It was first discovered by Dr. Lois Alzheimer in 1906 after he noticed changes in the post-mortem brain of a woman who died of an unknown illness. The brain examination led to the finding of amyloid beta plaques and neurofibrillary tangles throughout. These conditions have since been the pathological markers of Alzheimer's disease. I will be focusing on the amyloid beta plaques associated with AD. This is a cross-section depicting the physical deformation of the brain that occurs with the progression of Alzheimer's disease caused by the interneural tangles and amyloid beta plaques. So the amyloid beta gene, amyloid beta precursor protein, is found on chromosome 21, which codes for the amyloid precursor protein APP. A normal functioning amyloid precursor protein contains the growth-promoting properties and plays a role in the formation of nerve cells. The amyloid precursor protein is cleaved by alpha secretase, which forms the neuroprotective SAPP alpha family. But when cleaved by beta secretase, followed by gamma secretase, the amyloid beta is formed. These amyloid beta peptides, particularly amyloid beta 42, will then aggregate to form the infamous amyloid beta plaques that evoke neurotoxic events associated with AD. This diagram shows that the amyloid beta protein is seated in the cell by peptides 1 through 39, is cleaved by beta secretase followed by gamma secretase at peptides 40 to 42 to create A beta. So lowered A beta 42 concentrations in human cerebral spinal fluid is a biomarker of AD due to the aggregates forming in the brain. When paired with an increased concentration of tau in CSF, these biomarkers are conclusive of AD. A lowered A beta 42 concentration can be seen decades before AD begins to physically manifest. Here's a graph highlighting the decrease in A beta 42 concentration in CSF beginning 20 years before the onset of symptoms, paired with A beta deposition in the brain and increased tau concentration in CSF. So although A beta 42 is clearly diagnostic of Alzheimer's disease, it is extremely hydrophobic and is consequently extremely unstable in a lab setting. It is painfully difficult to try and accurately quantify A beta 42 from CSF. Just when aliquoting CSF using for quantification of A beta, a significant amount of A beta will be lost. Tests have shown that A beta can be reduced by 20% in the first 2 days, if not immediately stored in negative 80 degrees immediately after collection. It has also been shown that different polypropylene tubes can result in a reduction of A beta 42 by up to 62.7%. Recovery of A beta 42 is a focus of many labs and research because of how extremely difficult it is to control and quantify, which leads me to dry blood sampling cards. So dry blood sampling is a form of biosampling where blood is spotted onto filter paper. It was developed in 1963 for neonatal screening of disorders like PKU, HIV, and sickle cell. It is now routinely used in over 20 countries worldwide. All it takes is a small heel prick from an infant and a drop of blood to drop on this spot card for analysis. This test is superior to previous venous blood sampling because the minimal volume of blood needed, minimal training required to properly perform, and the increased stability with an ease of transport. This leads me to my hypothesis. I hypothesized that human cerebral spinal fluid can be safely and easily stored on traditional spot cards, and cerebral spinal fluid can later be extracted from the cards for precise amyloid beta quantification, overcoming pre-analytical difficulties. To test this, I have several experiments. First, method testing for loading the cerebrospinal fluid onto the spot cards, sensitivity of the cerebrospinal fluid on the spot cards. We will also test for repeatability, a range test for the standardized normal of A-beta 42 concentration, and a long-term stability of CSF on the cards. Now for my methods. Whatman filter paper cards were spotted with CSF, left to air dry, and stored at room temperature. Several methods to load CSF on the cards were assessed. For preliminary experiments to visualize the CSF pipetted onto the filter paper, 1 drop of food coloring was added to 1 milliliter of CSF. Internal standard was added to the samples at different points in the procedure, either before or after extraction in the development of the quantitative workflow. For identification and quantification, CSF was extracted using 5 molar guanidine hydrochloride and agitated for 40 minutes. The samples were then subjected to a solid phase extraction and elution and subsequently analyzed on a mass spec. We use this mass spectrometer because of its analytical specificity, superior to that of immunoassays or conventional high-performance weight analytes, and has higher throughput than gas chromatography mass spectrometry. This brings me to my results. For our preliminary testing, we attempted to collect the maximum amount of CSF onto a 6 millimeter punch of the spot cards. We succeeded in loading 100 microliters of the CSF only when the card is pre-treated and dried with 1 drop of food coloring, acting as a barrier for the CSF, not to spray, or when we allowed the punch to soak up the 100 microliters of CSF. So the first photo on the left is the CSF pre-treated with food coloring and then pipetted onto the filter paper, which only soaked up 50 microliters of the CSF. But when pre-treated with the food coloring acting as a barrier on the bottom left, it used all 100 microliters of CSF onto the 6 millimeter punch. These are the resulting chromatograms from our preliminary tests. Here we see an ion intensity of 1494.0 CPS for the A-beta 42 peptides. I compared the A-beta 40 peptides on the top because although they're not as conclusive of AD as A-beta 42 peptides, they're much less hydrophobic and prove that we can at least extract the CSF from the spot cards. So using the results from our preliminary test, I tested several loading conditions for our loading test. I compared a control of 200 microliters of CSF with punch circles that soaked up 100 microliters of CSF and dried, 6 millimeters of 35 millimeter strips that soaked up the CSF and dried, spots that were loaded with 1 drop of food coloring followed by a loading of 100 microliters of CSF and dried. We also then tested substituting 200 microliters of 4 milligram per milliliter BSA in PBS instead of water to give the peptide something to stick to. We also attempted pre-treating the cards with 20 microliters of internal standard with the food coloring before loading the CSF. And with the loading tests, I also tested a few preliminary dilutions to get an idea of the ranges of the sensitivity test. Here is the first round of results for our method testing. We detected less than 100 picograms per milliliter of A-beta 42 on all tests, besides the controls. Our best recovery of A-beta 40 was using the punch circles that soaked up the CSF, but only at a recovery rate of 5 to 6%. So using the data from the previous test, I decided to simply test whether we can extract A-beta 42 peptides from the punch circle cards. And doing this, I spiked in 20 times the amount of A-beta 42 peptides we thought we would see because of the 5% recovery rate seen from the previous tests. So using these spiked CSF, we now had a higher recovery rate of A-beta 42 than our previous test, even using the A-beta 40 peptides. Our recovery rate of A-beta 42 was a little over 30%. So I conclude that a maximum volume of 100 microliters of CSF can be absorbed under the filter paper punch of 6 millimeters. Adding dye to the filter paper interfered with the recovery of indigenous internal standard. Internal standard must be added following the extraction procedure. And based on the preliminary findings, this is a promising approach that we will continue to explore as an alternate strategy for storing cerebral spinal fluid and measuring AD biomarkers. A-beta 42 was also able to be detected between 2,000 picograms per milliliter and 3,000 picograms per milliliter, which is just above the 1,128 picograms per milliliter low A-beta threshold. So I would like to say that we will continue testing for sensitivity, repeatability, range, and stability of A-beta 42 peptides onto the guilty paper cards. I would like to acknowledge my PI and mentor, Dr. Mari DeMarco, for her guidance throughout the project, Grace for helping me running my samples, and other members of my lab for support. Thank you, and now I open the floor to any questions.